So we're continuing on with the signal transduction idea. And I just wanted to let you know I'm going to cut this lecture a little short because I have to get down to the Mead Auditorium. One of my grad students is presenting his thesis research. And so I have to be there to get him set up and introduce him. It's at 1 o'clock. It's right after this. It's at 1 down in the Mead Auditorium if anyone's interested. His project was looking at we used degraded soil down by the field plot down by Jacobetti, and we were experimenting with different organic and microbial amendments to see how the impact that would have on plant growth in very degraded soil. And so he's going to talk about that today. So if you're interested, it's in the Mead Auditorium, which is kind of kitty corner from where we are right now on the first floor of this building. Okay, so we left off here. And so I just want to kind of recap this idea that you have to have a signal transduction scheme in play in order for the hormone that is flowing to a certain part of the plant body. It will dock into some receptor, and then that tends to kick off this signal cascade. So on the left, we're looking at how bacteria do it. So microbes are the first living organism on the face of the earth. And so they have a scheme in place where they have a two-component, where they have a sensor kinase protein and then a response regulator protein. And just through a simple phosphate transfer from the kinase to the response regulator, you can get that signal cascade kicking off. And so plants have evolved from that idea, and they can kind of tweak it through the evolutionary process. So here you see that there's a middle protein involved in the cascade. But again, it's essentially the big picture idea is that you have phosphates being transferred as a way to start sending the signal into the cell and ultimately get some shift in gene expression. And so I'm not going to ask you to memorize the nitty-gritty of this. It's more that you understand it's taking the basic two-component, and then plants have evolved other mechanisms like a middle protein involved in the phosphate transfer. And then the next several slides look very busy, but again, there's some just big picture key things I want you to get from it, and the rest is just we won't have time to focus on. But here they're looking at 3 different ideas here with cytokinin hormone, the brassinosteroid type hormone, and abscisic acid. And so I'm going to take these one at a time. So here is cytokinin. And again, what's happening here? It's a phosphate transfer. All right, you don't need to know the names of the players other than I'd like that you know a sensor kinase and a response regulator. And then sometimes there's a middle protein or some extra proteins that are involved. So, but again, you can just see that there is this transfer of phosphate once the cytokinin is coming in and binding to the sensor kinase, okay? And then the phosphate transfer. And then you see, arrow down to, okay, the rest of the signaling cascade continues. So that's really all I need you to think about on that. With the brassinosteroids, again, over on the left, you see that it's waiting for its signal. On the right, you finally see the brassinosteroid structure there, the molecule binding. And you can see in this case, it's a phosphate transfer again, all right, to the target protein. And then they just say, onward, continuing on with the signaling cascade. So phosphate transfer. Here on the left is the absence of abscisic acid, and you can see what they're trying to show by this arrow with the perpendicular slash there. It's saying that is not happening, and so they're just saying it's inactive this protein is inactive. On the right, you see what happens when abscisic acid comes in and binds to the receptor. You can see that it will release this and allow for the phosphate to come in and bind, which again is triggering the signaling cascade. So slightly different version here. But again, the overall big picture is once you have the phosphate in play, that's helping to send the signal. Here are some ideas with a few different hormones. I've put in yellow the big picture idea, one of the big pictures here. Signal transduction pathways in plants often function by inactivating repressor proteins. So what that means is that there's a repressor protein sitting on the DNA repressing that stretch of genes, not allowing for transcription. And when the hormone comes in and binds to its receptor, very often what you see happening is, so let's look at auxin, figure A. You see the auxin molecule coming in, and what's happening is it causes that receptor protein to essentially come off or fall off. And it triggers the receptor protein. You see here, the auxin's actually messing with the receptor protein, binding with it. And that action actually causes the receptor protein to get chopped up and made useless. And so this is a slightly different idea, but again, the presence of the auxin hormone and the binding to the repressor protein is causing it to essentially fall off the DNA, come off the DNA. It's going to get degraded. And then the result is that you'll have transcription of those genes. Gene expression gets turned on with the result of that. And so they've mapped that out for auxins. They've also seen it for jasmonic acid and gibberellic acid. It's same basic idea here, where you have some kind of repressor protein making that stretch of DNA quiet, nothing's happening. But when the hormone comes in, it will bind with that repressor protein, which essentially causes that repressor protein to fall off the DNA, come off of it, and it usually gets chopped up and degraded. And then again, it allows for that stretch of DNA, the transcription factors start moving along and transcribing that stretch of DNA. So you don't need to know the names. Basically, what I just described verbally here is what you would be expected to know. It's the idea that the hormones are interacting with these repressor proteins in such a way to cause the repressor proteins to come off the DNA. And then that will allow for transcription to take place. So more and more, they're finding that this inactivation of repressor proteins is a common strategy with these hormones. So the next several slides are just taking these one at a time. But again, it's just a big picture idea that I'm looking for. You don't need to memorize names of these different proteins. And then I put this one here. I'm not really going to get into it, but I just wanted to make you aware that signal transduction pathways can get very complex. Okay, I've given you the general big picture idea of it. This figure is getting at how they feed into each other. So you can have multiple signal transduction pathways interacting to accomplish some result. And so that's what this is trying to show here, where you have, inactivation going on, you have augmentation going on. And so it gets complicated. And that's all I'm going to say about this. But if you are going on into graduate school and have any interest in some of the molecular biology of how organisms are doing signal transduction, it can get very interesting very quickly. Would, when you say, so it, so as soon as this repressor protein comes off, that's kind of like the signal, go ahead and transcribe. So so it's all context driven. It depends on, like, maybe this gibberellic acid is going to the seed and saying to the seed, go ahead and germinate. So then this would be genes that are going to get turned on to allow for germination. So it's really context driven on why is that hormone being sent to that particular part of the plant body. So it really does depend, but it's a pretty simple mechanism when you have that repressor protein interacting there. It just quiets everything down, and just by that their physical interaction here, it stops any transcription. And so then as soon as you release that, then the transcription factor just starts moving along and carries out the transcription. Kind of amazing.
So the signal works.
Right. Yes. So you're thinking about like seed germination, or maybe it's going to tell the stem, you know, we got to elongate. We're in the shade. Let's elongate. And so this might be a mechanism. Correct. So then maybe the repressor is saying, no, we don't want to elongate, or no, we don't want to germinate. It's the dead of winter. No. And then in spring, hopefully, you know, the gibberellic acid will say, okay, it's time to germinate. And so this flow would happen. So it's, and then, you know, I'm not a molecular biologist, but even so, I know enough to really appreciate how complicated this can get pretty quickly with plants, and how they have these multiple signaling cascade systems in play, which when you think of it, does make sense. Multicellular organism like us and plants, very complicated, lots of things that have to get coordinated, and it's very dependent on the habitat that the organism is in. Hopefully they're responding appropriately to the habitat so that they can adapt and stay alive and reproduce. So that's all I'm going to say about this slide. Okay, and then, so now I'm going to shift into the next slide set, which is still looking at hormones, but we're coming out of the signal transduction idea and more broadly saying, okay, what are these hormones doing for the plant, and some applied uses of hormones that we do when we interact with plants for a variety of reasons. So this is the applied uses slide set, and we're going to work through not all of the hormones, because I'm going to save some of them for when we get into plant defenses, which is coming up pretty soon here. So the applied use is thinking about how, now that we understand what some of these hormones do, how have we humans taken advantage of that when we are interacting with plants and working with them. And so auxins have a long history. You can use those as herbicides because it does mess. Too much of these auxin hormones can actually be toxic. You might have heard of the herbicide dicamba. That's kind of famous. 2,4-D is another one that's pretty famous. And so this has the ability, some plants are more sensitive to high levels of auxin than others. And so some can tolerate. So they found out that certain crop plants can deal with it pretty well, but then like the broadleaf weeds that get into the fields are more sensitive, for example. And so it just depends on the plant how sensitive they'll be, but certainly herbicides, hormones in excess often can be toxic to plants. And so herbicide use has been going on for quite some time. Okay, one famous idea that has been long known about auxin is that it allows for this property called apical dominance. So what this is saying is, here we're looking at a bean plant, and this picture got cut off, but at the tip of the shoot, the apical meristem, there's auxin being produced, and it's flowing down the stem. And what it's doing is, as it's flowing down from the apex and it's reaching the axillary buds where you can get new leaf and branches forming, it's telling, so you can see these axillary buds, they're there, but they're quiet, they're dormant. So apical dominance, auxins are flowing down the stem and they're telling all of the apical or the axillary buds region, be quiet, don't do anything, just hang out. If you amputate the tip of the plant, the shoot apex, the axillary buds will get released from apical dominance and they will start to grow. So you can do this with your houseplants, for example. If your houseplant's getting too leggy, getting too tall, and you don't like that, you can amputate essentially the apical meristem, and that will release, and the plant should start to, grow more in diameter. It'll get bushier rather than taller. So you can certainly play with that with some of your houseplants if you want, but that has been long known to happen. Yeah, pruning is certainly, when you prune, you could use this strategy. And then pruning also, of course, messes with the partitioning of sugars through the plant too. So you can, I was listening to a podcast, someone who owned an apple orchard, and they had a plant physiology expert on the podcast, and he said, you got to really aggressively prune your apple trees. And the owner was a new owner. He just bought this apple orchard. He's like, well, why would I want to do that? And the plant physiologist said, you have to prune it well enough so that you could swing a cat through the branches and it wouldn't hit a branch. I'm like, well, that's an interesting analogy. Swing a cat up into, I'm like, what? Caught my attention though. But he said you have to get that aggressive with, because it was a apple orchard that hadn't been maintained very well. And so he said, you really got to kind of prune really aggressively. And that's also messing with the ability of the tree to change its shape and not grow so tall as well. So you can influence very much the growth pattern of plants. Rooting compound that you might buy to propagate your plants. The rooting compound, that powder, sometimes it's a gel that you buy, and it's some kind of rooting powder title to it that usually has auxin mixed into it. And so you played around with that a little bit in lab, where we gave you different auxin concentrations. I think we tried gibberellic acid and kinetin as well. And then you had a control that didn't have any hormones added to it. You looked up with the mung beans to see if you had adventitious roots or not. And hopefully you saw in your control that you still got adventitious roots, even though you didn't spike in any auxin. And that's because the plant has its own auxin. And we didn't amputate the shoot tip. So there was plenty of auxin in the control plant. So they also should have formed rooting or adventitious roots. So I often don't work with rooting hormone. It's just an expense that sometimes I don't want to deal with, because I know the plant will have its own internal auxin that it will use to flow down to the roots and stimulate adventitious roots off the stem, I should say, of the plant. And so adventitious roots just means you're getting root production like off of the stem tissue. And it's not embryonic root tissue, really. You're essentially, when you cut a stem and you put that stem into water and wait for the roots to form, it's really quite amazing what's going on in the plant at the moment, because the cut stem of the plant now has to, essentially those cells have to go, oh, okay, now we no longer have roots. So those cut stem area, right where you cut it, those cells have to de-differentiate. They have to stop being stem cells and reorganize their gene expression to become roots. I mean, that's tremendous. They're totally de-differentiating and re-differentiating very quickly. You can often see plants responding and forming roots very quickly. It's really quite amazing. I mean, we can't do that if parts of us get amputated. It's not like we can just morph and grow in parts. So anyways, auxins are often mixed in to help promote that. And here, we talk a little bit about this in Plant Kingdom as well, but when you look at fruit development, and strawberries are the key one, you get this idea. So when you look at a strawberry, by the way, we learned this in Plant Kingdom, all the little so-called seeds on the surface of the strawberry, those are actually the fruits. Those are dry fruits. And the red part is actually the receptacle of the flower that morphed into that red, juicy, sweet tissue. So each one of these fruits is called an achene. It's an achene type of a dry fruit. Each of these achenes is producing auxin. And it's allowing that receptacle tissue to morph into that sweet, juicy strawberry. If you remove all of those dry fruits, the strawberry really doesn't develop. If you take that and then spray it with auxin, it develops. And so they were just doing this experiment to demonstrate you need auxins present during fruit development to get the mature fruit. So very much auxins are involved in fruit development. So another trick you can do is apply auxin early on into the early stages of flowering, and many plants will dedicate and just form female flowers instead of like a perfect flower, for example, or you can get them to shift and form just the female flower. And so they've played around with that with a variety of different plants and have discovered that some plants will do that. You can spray your plants also to fruit set just means the final stages of, well, or I should say the fruit development process. So if you spray your plants, you can get fruit development without seed production. That sounds weird. But I'm sure you've eaten seedless oranges, for example, right? Seedless watermelon, bananas are pretty much seedless. So when you talk to a botanist, you say, well, how do fruit form? Usually they say, well, in order to fruit form, you have to have the sperm uniting with the egg in the flower, which creates the seed, which then creates the fruit. And so that's the normal way of how we get fruit. Here we're saying you can get it and just bypass the seeds if you spray with auxins. And so that's a pretty potent impact on the plant where you can get fruit without any seeds in it. So that the fancy word for that is parthenocarpy. The common way to say it is seedless fruit. So that is a pretty profound impact on the plant life cycle. So they can take use of that for seedless oranges, for example, big business. Yeah.
Like bananas.
So banana production, when you look at banana plantations, it's the banana plant comes up from like underground rhizomes every year. And like the bananas we're eating currently, your parents ate a different type of banana and their parents ate a different type of banana because we've had plant pathogens. They've been sequentially wiping out our banana plantations over the decades here. So like a student that I worked with in undergrad, Colin, he gave me a baby banana plant, and it's actually a banana plant that my grandfather would have eaten, or it would produce those bananas. It's an heirloom banana plant essentially that we have in the greenhouse right now. At least I hope it's still in there. It was happy in there a few weeks ago, but anyways, yeah. So bananas, when you see their plantations, they just grow those plants by asexual, they sprout up every spring from underground rhizomes. So. Agent Orange has a famous reputation during warfare. That was during the Vietnam War. Agent Orange was used. They would fly planes over the forests and spray Agent Orange onto the trees, and the leaves would drop. And so it essentially is producing, it's creating a situation that kicks in leaf abscission, essentially. And that we already talked about leaf abscission, how it is so much of it, it actually triggers ethylene and causes leaf abscission. So it's like a toxic level of auxin, and the plant kind of freaks out, starts pumping ethylene, and then that leads to leaf abscission. And they did that during warfare so that they could see the enemy better and do more killing. So. Sometimes you want to more precisely determine when you want that fruit to come off the mother plant. So if you're growing fruit for your living, sometimes you want to carefully control that. you can actually spray auxin as an act of pruning rather than physically cutting. You could actually spray chemicals and get the same impact. And so some people choose to do that. We talked about the herbicides already. The dicots or the broadleaf plants tend to be more sensitive to that. So the ones that are popping up in your field, if you have a cornfield, the corn plants can handle the herbicide. Any of the weeds in there tend to die off from it. And then the rooting compounds we talked about and the seedling fruits. I just kind of put everything onto one page here, onto one slide. So auxins are probably the most well-studied group of hormones. They were the first to be discovered, and there's just been a lot of activity in auxin. You know, what does auxin do for the plant? How can we make use of this? And auxin, like I said, they have yet to create a plant that's a mutant for auxin production. So auxins are like, plants have to have it or they die. Same with cytokinins. It's the same idea that cytokinins, they haven't found a mutant plant yet that can be deficient in cytokinin. Again, they can be used as herbicides. The cydiazuron chemical is an herbicide. Zeatin is what the plants make. That's their version of a cytokinin. And then we have plenty of synthetic cytokinins that we can make in the lab and expose the plant to. Cytokinins are very commonly and heavily used in cloning of plants. And so when you want to clone a plant in tissue culture techniques, you have to work the ratio of auxins and cytokinins, and it's very specific to the plant you're working with. So you have to get to know your plant and figure out what level and ratio of these two hormones do you need, because you need the correct balance in order to promote, first of all, just cell growth. You want, so this yellow part that you're seeing in the image, it's kind of yellowish part, that's the callus. The callus, and you're going to be learning about that this week in lab tomorrow, right? Tomorrow's Thursday. So you're going to be learning all about this. You're actually going to take a crack at cloning plants yourself. So the callus is just undifferentiated plant cells. And then with the right ratio of auxins to cytokinins, you can trigger those callus cells to start to differentiate. So some cells will devote to becoming shoots and leaves, some cells to roots, and you'll get an actual baby plant that you can nurture, you know, originally from those callus cells. So it's really the ratio of those two, and it depends on the plant what those ratios will be. You got to play with it a little bit. So this is big business, like all the African violets that you see pretty much for sale. It's usually done this way now. They mass produce through plant tissue culture techniques. It's much easier. It's you can stay consistent. Once you find the perfect African violet plant, you can just clone it over and over using these techniques. And so most of the African violets for sale are done that way. Here you're looking at lettuce, some looking better than others. So here you're looking at transgenic lettuce plants. So they've here you can see the ones on the bottom. It says that lettuce plants expressing the IPT gene. And IPG gene in the notes I have down there, the gene involved in cytokinin biosynthesis. All right, so what this is doing is they kind of beefed up the lettuce's ability to produce a good quantity of cytokinin. And so it's telling the plant, we're healthy, don't senesce, don't die. Let's keep living, let's keep functioning. The ones up here were the control. So these are producing and continuing to produce cytokines and keeping the leaf tissue healthy. And so that's a strategy that can be employed. They've also found that with rice, which is big business, you can pump up the amount of grain per plant with the correct application of cytokinins. So these two genera of rice cultivars, the Japonica and Indica, they've done studies with those, and what they've found is with the Indica variety, they have a situation, cytokinin oxidase actually breaks down, that's an enzyme that breaks down cytokinin. So if you don't have that enzyme very active, that means you have still plenty of cytokinin, which means the cytokinin is there saying don't senesce, keep producing grains. And they actually see as a result, it lives longer essentially and healthier, and it ends up producing more grain. So just tweaking, messing with that enzyme that breaks down cytokinin is a strategy that can be used. So here's some images. Whoops. Japonica and Indica here. And they're showing a close-up of the grain yield. So they can use this idea, get more. I think you can see that Indica has a nice grain production there. So they can get a better yield, which of course is going to give you a better harvest, more money from selling that. The giant pumpkins, you said? What? I think the one that won this year, you know that they have giant pumpkin contests, right? Yeah, I thought it was thousands of pounds this year. And I think somebody in Minnesota was the winner this year. I'm pretty sure. How do they do that? Good question. I would love to know what are your pumpkin sources? Where are you getting that from? Because I mean, they're massive, just incredibly massive.
And how do you even make it?
They actually put it into this big like hammock harness and put it onto like, you know, those weight, those balances like semi trucks go onto, you know, that kind of thing. Did you know that they do pumpkin regattas where you carve out pumpkins and float in them? Growing? Oh my God. 1,700 pounds. That's dedication to keep that thing going. Oh my gosh. Yeah.
Now you have to use
Yeah, they get deformed because of the physics of the weight, you know, the gravity. They've looked into that too, like the physics of giant pumpkins. And I think so. I don't know what they do with them. Yeah, I don't know. I've never seen the inside of one.
But they don't have a lot.
Oh, really? So the resources that have been kind of devoted too much into the fruit. Interesting. Yeah, I bet you could get a pretty penny for those seeds from people who are into, you know, next year I'm going to do it. I'm going to grow the biggest pumpkin. But I like the idea. Lake Superior right here, we could do pumpkin regattas. I think that'd be awesome. You have to wear a wetsuit around Halloween, but they usually do it around Halloween. Has anyone seen the movie, Wallace and Gromit movie, is it Curse of the Werewolf with a pumpkin? Gromit's the dog, right? And he grows giant pumpkins. And Wallace and Gromit, do you know what I'm talking about? It's hilarious. You got to watch it. I think it's Curse of the Werewolf, right? Is that the one? Were-Rabbit, Curse of the Were-Rabbit. I have it on DVD if anyone wants it. It's awesome. I watched that over and over with my boys. It was hilarious. I still watch it, just me, because I like it. Just to be aware, if you are perusing shelves of beauty products, for example, you might find plant hormones being used in that regard. I always find that very entertaining when I look at some of the skincare things for sale. This is just one example. I'm seeing more and more of it now. Kinetins are being put into creams. Resveratrol, which is a secondary metabolite we'll talk more about. Red wine extract. Another one's sephenol. And so they're getting into this idea of, you know, because they know that cytokinins can promote cell growth and stop kind of the aging in plants, right? It delays senescence in the plant if you have a flush of cytokinins. So they're putting that into human beauty care products. Does it work? I don't know. I haven't read. I'd like to see, like, where's the data on that? Show me some control versus treated. And I have not actually tracked that down. So I don't know. But be aware. Yeah, I'm sure these are expensive. I've looked at some of the prices online, you know, when I was hunting for some of these products, and of course they sell them at a high price because they are getting you to believe that it will work. And I don't know if it does or not. I'm not saying it doesn't, but I don't know if it does either. So, all right. Ethylene. So remember ethylene. I want you to know this part of the pathway. It's the Yang cycle, but I just want you to worry about the last few parts here because this is where they've been doing a lot of research on controlling levels of ethylene. And so you can mess with these enzymes in order to get that to happen. If you want to decrease ethylene, you could put the plant product. So very often what they do is they store harvested food products in like rooms that are controlled for the atmosphere, and they'll lower the oxygen level in the room or in the container, and they'll pump more CO2 in there and to try to minimize ethylene production. Because once you harvest the apples or tomatoes or whatever, I mean, those cells are still living, so they're still doing physiology, and so now they're separated from the mother plant. They're going to start to senesce over time. It's just the nature of it. And so if you can try to stop ethylene production, that can help prolong the freshness. The flower industry, the cut flower industry, huge business, billion-dollar business. And again, they take advantage of that by, they know that they need to block the ethylene production. And so the left flowers were soaked, the stems are sitting in a solution of thiosulfate. And then control plants looking pretty pathetic. I don't think you'd want to give those to anybody. And that's just after 14 days. So the cut flower industry, they're certainly aware of this. At least at some level, they know that they have to make sure to get the right chemicals that the cut stems are soaking in to try to block that. So having said that, you know, you still get, as soon as you cut flowers off the mother plant, you're creating a wound, right where you cut it. So right away you got ethylene going because you wounded it. And so you got to try to like sop up the ethylene, and there's chemicals that will do that, kind of like stop that ethylene. Well, here, it's the next slide. So you have inhibitors of ethylene action. So you try to flood the receptor with a different chemical so ethylene can't get in. So there's competitive inhibitors that they know can dock into the receptors and take the place of ethylene, so then ethylene can't send the signal. So that's a really simple good. High CO2 levels around the cut flowers or around whatever. Adding silver, Ag+, adding silver into a solution will help when you put the cut flowers into that. You can try to inhibit the production of ethylene. And so you can mess with this ACC synthase. You can absorb the ethylene. So potassium permanganate is a chemical that will react with ethylene and kind of sop it up so that it doesn't have a chance to act on the receptors in the plant. So again, if you're storing food products before they make it to market, you have to be aware of these strategies or you're going to lose your whole profit margin. They're all rocked and will be done. Okay. I think I'm going to stop here and we'll continue on with this Friday. We'll keep looking at some hormone uses